Good evening, everyone, and thank you for joining us. My name is Dr. Jeremy Steinbacher, and I'm the Director of Operations for BioInspired Syracuse. We are so happy to have about 100 faculty, staff, students, alumni, and other friends of the university join us tonight for a look at COVID and the work that some of our esteemed faculty are doing to investigate it. I'm going to turn things over now to our host this evening, Dr. Lisa Manning, who's the Kenan Professor of Physics and the founding director of BioInspired Syracuse. Lisa?
Thanks so much, Jeremy. Hi, everyone. Like Jeremy, I'm so excited to see so many of you here for our public lecture and panel about COVID-19 research at Syracuse University. As Jeremy mentioned, I'm director of the BioInspired Institute, which is sponsoring this series of public events. The BioInspired Institute is a relatively new research institute in Syracuse, founded in 2019. It focuses on interdisciplinary research at the intersection of materials and living systems, which means we're interested both in developing new types of bio-inspired or biocompatible materials, and also in using the lens of mechanics and movement to understand how living organisms function and how that function breaks down in disease. Therefore, our mission is to support research into complex biological systems, as well as to design and build smart materials to address global challenges in health, medicine, and materials innovation. In this type of interdisciplinary research, where people are willing to step outside their comfort zones and pivot to new problems that allow scientists and engineers to tackle big issues together. As I think you'll see during the course of our program tonight, our researchers who work at this intersection of materials and living systems are able to suggest surprising and powerful hypotheses about what drives the novel coronavirus. I think because they think outside the box and they have a skill set to work on these highly interdisciplinary teams. It is a significant understatement to say that this year, and specifically the COVID-19 pandemic, have been extremely challenging for all of us in different ways. Maybe some of you, like me, have young children and are trying to balance work with continued school and daycare closures. Many of you are likely worried about older family members or those with special needs. Faculty researchers here at Syracuse University have been dealing with those issues too, while also trying to protect jobs for graduate student researchers and switching to teaching courses in hybrid and online formats. For me, one of the very brightest spots in all of this mess has been watching how a significant fraction of our researchers at SU, despite these challenges, they have fought to figure out how they can address the pandemic. Given their extensive scientific training, they have scoured the new research findings about COVID as they come out to find places where their area of expertise might be useful in combating the virus. So tonight, I am very pleased to moderate a discussion with 3 faculty members at Syracuse University who have significantly altered their research to focus on COVID-19. And they have already achieved some really exciting preliminary results. We'll first hear 2 short presentations by the faculty members highlighting these fundamental science research with a goal to understand and hopefully identify new targets for antiviral therapies. After that, we'll turn to an interactive question and answer session. So please feel free as these presentations go along to enter questions into that Zoom Q&A box, and we'll select some of those to address during the question and answer time. So first, I will formally introduce our faculty panelists, and then I'll turn it over to them because they're the superstars of the show. Okay, so first I'll introduce Professor Alison Patteson. She is an assistant professor in the Department of Physics and a leader of the Mechanics of Development and Disease Focus Group in the BioInspired Institute. Dr. Patteson's research aims to identify physical forces that shape life. Her group uses tools from engineering, biology, and physics to investigate emergent collective behavior of living systems. Dr. Patteson received undergraduate degrees in physics and mathematics from Kutztown University of Pennsylvania, a PhD in mechanical engineering and applied mechanics at the University of Pennsylvania, and she continued research as a postdoctoral fellow in the Perelman School of Medicine at the University of Pennsylvania. She joined the faculty at SU in 2008. She won a very prestigious dissertation award given by the American Physical Society Topical Group on Statistical and Nonlinear Physics. And this year alone, based on high evaluations from her scientific peers, she has been awarded 3 different grants from the National Science Foundation to support her fearless and exciting research program. We're really lucky to have her here at Syracuse. I'd next like to introduce Professor Jen Schwarz, who's an associate professor in the Department of Physics and a member of the BioInspired Institute. She earned her PhD in theoretical physics from Harvard in 2002 and continued her research as a postdoctoral associate at SU and then UCLA and then the University of Pennsylvania. She returned here in 2005, signing on as a physics faculty member, and has been here since. She is an expert in the mechanical properties of disordered materials, and she's becoming increasingly interested in modeling biology from a physics perspective to understand how the brain works and how viruses enter cells, as you'll soon see. She has received several departmental teaching awards, an NSF Career Award, and most recently, an Isaac Newton Award for transformative ideas during the COVID-19 pandemic from the Department of Defense. Like Isaac Newton did during the Black Plague, she lives in the middle of an apple orchard, and she hopes to use apples for physics inspiration in ways that Sir Isaac Newton couldn't even imagine. Finally, I'd like to introduce Professor James Hougland, sorry, Jimmy, is an associate professor of chemistry at Syracuse University and a member of the BioInspired Institute Executive Committee. He directs research investigating the chemistry and biology of proteins that are first created and then later modified by the cell to generate new functions. His research group studies proteins that are modified to stick to membranes at the edges of cells, which are important in many different types of human diseases. And his research program receives support from multiple funding agencies focused on human health, including the March of Dimes, the American Diabetes Association, and the National Institutes of Health. After growing up on the banks of the Mississippi River in Western Illinois, Dr. Hougland received his undergraduate degree at Northwestern University, and then he got his PhD in biochemistry from the University of Chicago and was an NIH postdoctoral fellow in the chemistry department at the University of Michigan before becoming a faculty member here at Syracuse in 2010. Dr. Hougland also holds a courtesy appointment in biology at Syracuse and an adjunct appointment in the Department of Biochemistry and Molecular Biology at SUNY Upstate. So I know that was a bit of a long introduction, but I wanted to show you how qualified and fancy pants our speakers are today. And without further ado, I will turn the mic over to Professor Patteson and Schwarz. Thank you.
Thanks. Thanks, Lisa, for the introduction. And also for the invitation to speak today. This is great. And to Jeremy and others who helped set this up. All right, let me share full screen. Okay, so the title of my talk today is How Physics Can Help in the Fight Against COVID-19. And I hope I can answer this question in part and also be able to tell you a little bit about some of the things we've been doing in the lab. All right, let's start on my timer. Okay, but first, I want to make sure I acknowledge everyone who's helped out on this program, because this work wouldn't be possible without this really great dynamic and interdisciplinary team that we have working on this project. Oops. Can I not go back? Let's see. Okay, back, thanks. So first is Max, who's a graduate student in our physics department, and also Sarthik. We'll be hearing about their work. And Danielle, who's an SU physics undergraduate. And I also want to just acknowledge our collaborators, Robert Buczki, who's in the microbiology department in Poland, and also Paul Jamney at UPenn. And I'll also touch upon the work we're doing with industrial partners, Integral and Nascent, and you'll see their names pop up later. Okay. So first, let's try and set our mind into the right context and what is the world that a cell sees or a virus sees. And so this is a nice link that actually will hopefully pop up in the chat. It shows you kind of the scale of a cell. All right, we know like what a grain of salt kind of looks like. We can kind of see that. or a human egg. And look, there's the cell, that's a skin cell. And we keep going. We see a little yeast, there's a bacteria. We're going down now into nanometers. And there's an HIV virus. That's actually about the size of the COVID molecule. Here we're getting into some very small molecules. And now we're down to water and a single carbon atom, which we thought were the smallest molecules for a very long time, the smallest particles. So zooming back out, there's the virus. There is the cell that the virus infects. And that's what we're going to be touching upon. So we're all these different length scales, and this is actually a place that physics is really able to kind of scale different sizes and look at different physics that actually occur at these really small length scales that we don't see. These topics will come up. Okay. All right, so first I want to kind of just touch upon the primary way that this virus is transmitted. Okay, so, and actually there's even some physics here. So the virus is spread through respiratory droplets. And these droplets come in a wide range. We don't see them. Some of the ones that are kind of, they're about on the order of your hair or smaller, the diameter of your hair. So some of the ones that are on the larger end, the diameter of your hair, fall projectile motion. They fall straight down to the ground pretty quickly. But some of them are very small and can be dispersed out from 3 feet, 6 feet, and actually beyond. Each of these droplets that you see here in the little schematic, they hold many virus particles in them. And just, you know, for scale here, if this is the diameter of your hair, a respiratory droplet is this many times smaller. That's containing many of the viruses. Okay, so why is this important? It takes us to our first tool in preventing spread, which are masks. So masks are designed to trap those respiratory droplets from spreading. All right, and your masks, your typical cotton mask is made up of little fibers. If you zoom in, you can see there's little gaps. The gold standard is the N95 respiratory masks. And what these masks do is they take those respiratory droplets, right? And they catch them because the fibers create this mesh the mesh of the fibers is smaller than the particles. So they get stuck there. Again, for reference, this is like the diameter of your hair, the small droplets that get stuck. And this tiny little point there, that's a virus relative to scale. So the virus is small compared to the mesh, but most of those are pretty much, basically all of those virus particles are trapped in respiratory droplets, which will get trapped in this mesh. All right, so one thing that actually fluid dynamicists, engineers in physics who study fluid dynamics, can actually very scientifically test how effective these masks are. So this is actually a test. There's a laser sheet here, and there's some fluorescent particles labeling droplets that are the size of your respiratory drops. And here, if you have your cotton mask, you can see there's very little that kind of gets through. Some, but not nearly as much or as far as if you're not wearing your mask. And actually your mask is designed to protect you from others. I think I have my title wrong, sorry. But your mask is not designed to protect you, really, it's designed to protect others. So in this little schematic here, right, if you're not wearing a mask, these respiratory drops go out into space. And if someone else is wearing a mask, they get some protection. But really, you get maximum protection when both people wear masks because you can filter out just maybe the tiny few that are able to get through the masks. All right, and you know, scientists have these fancy tools because they have, you know, fast cameras and things. But actually, you could try this at home, and you could ask, how effective is my mask? Actually, all you need to do this is your mask and these things called agar plates, AGAR. You can actually buy them on Amazon. They're pretty cheap. I think you could also maybe use some leftover food, but that might start smelling. So what you can do is you place these plates out, say at different distances, and you open up the lid, and you can breathe. You could pick a condition: breathe, cough, sing for like 15 seconds or some time period. And then you can actually kind of see how many of these, how much of this projectile comes out from your mouth, because when those droplets hit the agar, not only do they have things like viruses, but they have lots of bacteria, which is, yes, we're gross animals. That's why we brush our teeth every day. But those bacteria, when they land on this nutrient-rich plate, they're very happy. They like the conditions. They start growing and growing. And actually, I guess depending on how warm your house is, and the next day, you'll see a colony where there was a respiratory droplet and where there was bacteria. So you can actually test, for what distance and which condition, mask or no mask, has more exposure to potential pathogens in a viral load. And actually, I don't know if we're set up to do a poll, so I'm not sure if I get live feedback here, but ask yourself, if you were going to the store and you were to interact with someone, would you want to be in the condition on the right or on the left? Right? If this is all the things that you could be breathing in. And I think most of us would want the condition on the left, where there's very few droplets that are being exposed to you that you could possibly breathe up. Now, what scientists will do is they'll do this dozens and dozens of times. And, they might catch a few of these droplets and they can actually calculate the statistics. And we know now that actually masks are very effective. If both people are wearing masks, compared to, I guess if both people aren't wearing masks, I think it's about 70% chance of infection for close contact. But if both are wearing masks, it goes down to 1%. So we really have a lot of control over how much this virus spreads. Okay, let me see. All right, so let's suppose you do get exposed. And whether you're wearing a mask or not, someone coughs and some of those droplets make it up. You breathe them in. What they're going to do is they're going to travel down and into your lungs. All right. And your lungs actually have these alveoli capsules, and that's where they first see your cells. Okay. So this is inside one of those capsules. This is one of the viruses. This is some of your neutrophils that want to actually fight against those viruses. But this is the first chance that the virus can enter into your body. And there's two main ways that cell can infect and get into your cell and start wreaking damage. So the first on the left is fusion, and the one on the right is endocytosis. I'll just briefly describe these. So in fusion, the virus here interacts with a receptor. That's the kind of key here. It has to actually bind to the cell, and it has to have a specific kind of interaction to do that. And there's a nice animation, which I'm gonna let play here. There we go, of this happening. This is actually HIV virus, but it's very similar to how the coronaviruses operate. So that's the receptor. Those are kind of the spikes outside the cell. And those kind of spikes act as these hinges, which like pull the virus membrane into the membrane of your cell. So in this case, the virus is on top and the very membrane of your cell is on the bottom. And once it can do that, those layers combine or erode, and you see it's gonna able to inject its DNA, and then it's kind of game over. Okay, at that point, the cell has the viral DNA and it's going to start replicating that viral DNA, which creates more and more of the virus inside your body. On the right, we have endocytosis, and Jen's actually going to tell us about that in a little bit. So I'll skip over that for now. Okay. I need to speed up a little bit, but I want to just tell you kind of between March and today, where we are, because, the first thing that made me really notice, although I was aware of the COVID-19, was on March 1st, I got this headline that March 1st, actually, I woke up because I was supposed to go catch a plane to Colorado to attend the APS March meeting, which is the biggest physics conference. And I woke up and it was canceled. I was not supposed to get on the plane and go to Colorado that day. Okay, so we were realizing this is serious. And then, next few weeks, Tom Hanks has coronavirus. It's spreading. Soon, cruise ships don't know where to dock. Now the healthcare system's overrun, people can't get tested and people can't get ventilators. Right? So this is very serious. And actually we're in this climate actually again, unfortunately. So back in March, reading these headlines and learning about the coronavirus, this novel coronavirus. So I'd like to just, plug that the first coronavirus was discovered by a woman in 1963. And this new coronavirus is due to a mutation that occurred in just like 12 genetic letters of the DNA of this virus. Okay, and this mutation is thought to actually help the virus bind to our cells more tightly with a receptor. It's kind of that schematic there and why this is potentially more dangerous. And as I was reading more about this new virus, I came across this headline, which is surface vimentin is critical for cell entry of SARS-CoV-2. And maybe this is sounds very scientific and jargony, which is fine. But what I noticed was this word vimentin, right, that title, because vimentin is one of the expertise of my lab. That's what we study. We study the biophysics of this protein. So vimentin is part of the cell's cytoskeleton. It's actually a rather obscure protein that we study. Actually, as a physicist, we like to study how stiff it gets when you stretch it. This is a vimentin sample. This is slime that you can pick up at Target, but it acts very much like vimentin is if you can purify it out of the cell. And so we're very interested in actually how this slimy protein gives the cell its strength and protects it from damage. But we thought, okay, we have a lot of tools to study this protein. Let's see if we can address a question about whether it's involved in the new this new SARS-2 virus. All right, so in this framework from this paper that we learned, that I was studying, we found that vimentin actually can be on the outside of the cell, where normally I only look at it inside, and it seems to act like these sticky sites where the virus can bind. And I asked this question, well, can we prevent cell uptake by preventing this interaction? So how would we do that? Well, there are drugs or things called antibodies that can stick to different proteins. In our case, we want to stick this drug to the vimentin. And if we do that, then the virus can't stick at vimentin where it wants to stick. And actually, we're able to, there are these molecules available. We had some in the lab, and actually there was one that was FDA approved from Nascent that we decided to try and block this interaction. So the experiment looks like this. First, we have to infect the cells. So we're using a virus-like particle. So it is coated with the spike-2 protein, but it doesn't have the viral DNA in it. Instead, it has a GFP reporter, meaning that if the cell uptakes it, it starts expressing this GFP, it starts fluorescing green, which we can see. And then we compare to a test case where first we expose the cell to the drug, allow the drug to cover the cell surface, and then expose it to the virus and see if it reduces the number of GFP molecules. All right, so this is what our control case looks like. Every little green dot you see there is a cell that's expressing GFP, and it's a cell that's been infected. And then we try a drug, and we look at how many we have, and then you can look at the slide and see which one has more green cells. It's the one on the left. So our drug, our vimentin antibody, was able to block uptake actually by over 50%. And of course, what we do is we count actually how many cells are infected by how much of the drug that we add, and actually lots of controls and lots of iterations. And we're finding that you can actually block more and more of the viral uptake with more and more of the antibody. All right, and then this was the time that we celebrated. Now is the time of COVID, so we're social distancing, but in the words of my graduate student, Max, mission accomplished, pop the bottles. Okay, all right, Jen is now gonna take over. Oh, we can't hear you, Jen.
if we were to zoom in on a cell, a little green cell, an infected cell in one of Professor Patteson's experiments, we'd want to ask, what is happening? So again, if we look at different scales, we have the scale the size of the universe on one end, the size of a atomic nucleus on the other, and in between we have again the size of the cell, around 10 microns, and we have the size of the virus, around 100 nanometers. And so in other words, we want to ask, how is the virus getting inside a body to potentially wreak havoc? And so, as Professor Patteson mentioned, we're going to focus on now endocytosis. There are two ways the virus can get inside the cell. One is membrane fusion. She showed you that really cool movie that looked like a monster invading a cell, pulling up the cell membrane. Here, this is a schematic of the cell membrane really deforming and engulfing and eating the virus. This process, in a past life, I guess I had thought about endocytosis because endocytosis is just a generic way for how cells eat things. They not only eat viruses, they need to eat other things to stay alive. And so in thinking about endocytosis, here is the outside of the cell on the upside and the inside of the cell on the downside. And so you can have these large invaginations or shape changes in the membrane. And so I guess in a past life, I ended up proposing a new, what's called pinch-off mechanism, so that this droplet here can then pinch off and go inside the cell. So now, if we think about the problem at hand, how a virus invades a cell, we can ask, let's have a hypothesis for what's happening, since we don't have an actual microscope at hand. So let's say, okay, vimentin, the magic protein that Ali talked about, vimentin binds to the cell membrane, and it also binds to the spike protein. And let's say in doing so, vimentin helps the virus be wrapped by the cell membrane. And since we know many of the rules of interaction between the different players in the system, we can actually run a simulation to see what happens. And so here we have the cell membrane in blue, the vimentin in green, the ACE2 receptor, it's a receptor for the virus in purple, and the virus in red. And so now we're going to run a simulation and see what emerges, some collective phenomenon. And this was run on some computers in the Green Data Computing Center at Syracuse. And the coding was done by a super talented graduate student, Sartik Gupta. So next, we're going to see now what happens when we know the rules. Let's run the movie. And
I'm sorry, Jen.
That's okay.
I'm not sure why it's not running.
Okay. All right. Well, if you were to run a movie, you could see that the blue would be wrapping around the virus particle. Okay. All right. And this is actually a 2D simulation. We're now testing this in 3 dimensions, a more realistic case. And so stay tuned to see what happens in 3 dimensions. In 2 dimensions, we do see the wrapping, the endocytosis that is facilitated by vimentin, and we'll now explore how it happens in 3 dimensions. So with modeling, we hope to quantitatively explain the 50% reduction in the SARS uptake with the vimentin antibody. We hope to also make new predictions to be tested so we can really understand what's happening again at the scale of the virus, what the virus sees. And with this understanding, we can help stop viral uptake.
All right. Thanks, Jen. And just to quickly wrap up, we've been able to, seems like, identify a mechanism by which the virus can enter the cell and possibly a way to block it. And there's applications here. We can take these antibodies and perhaps, for instance, put them in a nasal spray that you could deliver, which might actually prevent you from taking up more of the virus if you were to be exposed. And also remember, still our number one tool to block COVID is to wear a mask, and only you can prevent the spread of the virus. So that's our slides, and I'm now going to pass it off to Jimmy.
Okay, here we go.
Thanks.
There we go. Thank you very much, Ali and Jen. And thank you everybody for coming tonight to hear about some of the work we've been doing here at SU, aiming at figuring out different ways to kind of get in COVID's path and try to make things a little better. So I really appreciate it for the introduction to genes and DNA and proteins, because that's really an area where my laboratory works. In particular, we study this process called post-translational modification. Now, what this is as we've seen Ali describe, right, we have our genes, which sort of code for proteins. So if you think by analogy to a car, the genes are the plans for building a car, right? Then our bodies make the protein, so you build the car, right? And you have a little bit more, right? So if you go out, there's one plan, you can get the 4 different models, you know, the regular model, the Grand Touring with the leather, you know, right? You have a few different options in our cells as they make the proteins from our genome. What my lab studies is then subsequent chemical modifications that are done after the protein's made, right? So if the genes are the plans for the car and the proteins are the 3 or 4 models you can buy at the dealership, post-translational modification is where you get to put on the roof rack and the chrome and the spinning rims. Okay, so you really get to customize the protein. And particularly in my lab, we like to study how proteins are chosen to be modified and then what effect those modifications have on how the protein works. In particular, we focus on a subset of modifications, which are called lipidations. And I'm showing this here. I often joke, so I am a chemist by training and I'm in chemistry department. So I feel contractually obligated to show a picture of a chemical at some point in the talk. What we have here is a protein where we're attaching this big, long chain of carbon atoms. Really what this is basically fat. It's going to make the end of this protein more greasy. And one of the things we study in my laboratory are how are proteins chosen to be modified in this way. And I want to highlight the work of Siddharth Ashok, who is one of my former grad students who did a lot of work to set the stage for what I'm going to tell you about. Bring out Siddharth, he actually defended his thesis this summer during the pandemic virtually, and now he's in med school at SUNY Buffalo. So Siddharth's moved on to the next stage of trying to help out people with this, with our pandemic response. But when we look at lipidation, as I said, we're putting on this greasy group here. And what they can do very often is take a protein that wants nothing to do with cell membranes, like the membranes we just saw Ali and Jen talk to us about. By putting that lipid on, now the protein will interact with the membrane. So we've been able to change the area code. We take the protein from floating around everywhere and we stick it into the membrane. And as Lisa said, this is something my lab studies a lot. Now, we've usually been focusing on human proteins, but when the coronavirus came out, my lab, like many other labs, started looking to see how could we contribute to the effort to try to deal with this. And so I started looking at the genomes of different coronaviruses. So here I have 4 known human coronaviruses. These were known beforehand. These all basically cause the common cold. So there's a good chance everyone in the audience has had one or more of these coronaviruses. In this, each box here codes for a different protein, and this diagram is showing us the whole genome, all the proteins that are coded for by each virus. Now, when we look at these new members of the family, we have COVID, and then we have SARS, which if you remember, came around in the early 2000s and caused an earlier pandemic. SARS and COVID are very highly related. And one thing we notice is this one protein, 7B, that seems unique to these 2 proteins. And so this got us interested in looking at those proteins and saying, is there anything we can learn about how they're modified? Give us a target, give us a handle that we can attack to try to muck up virus function. And when we zoomed in on this, I'm going to call it ORF7B, but just 7B, we noticed that the end of the protein had a particular amino acid sequence that Siddharth's work had shown about 1.5 years ago in human cells could be modified, could be lipidated. That would mean that this protein might actually interact with cell membranes. Now, we believe that some of these coronaviruses use these accessory proteins to improve their infectivity. And so it led us to a question that we wanted to answer. Is this ORF7B, is this protein lipidated? And so we built up a hypothesis, right? Scientists, we love using if-then statements, right? So if the protein's modified, and if that modification is required for the virus to work really well, then if we block the modification, it gives us a new way to block COVID, to treat the disease. And so we wanted to explore this using tools we have in my laboratory. So one way we want to do this is say, when we lipidate proteins, they tend to like to stick near cell membranes. So let's explore that in a model system. This work I'm going to show you was done by my my lab manager, Michelle Seberg. Actually, during the pandemic, we got clearance to come back in when the labs were closed to start working on this project. And we started using this same protein Ali mentioned, GFP, green fluorescent protein, which is nice that it lights up and lets us see where the protein is in the cell. Now, if you put GFP just by itself into cells, here's a picture of a bunch of cells in cell culture by visible light. Here's fluorescence, and you'll notice the GFP is basically everywhere. In my lab, we call these jelly beans, right? No localization, it's all throughout the cell. But then we said, okay, we know we can light it up, but now let's see what happens if we force GFP to hang out near membranes. And we did that by attaching GFP to another protein we study in my lab. It's called KRAS. It's involved in cancer signaling, but that's not important for here. What's really important is we know KRAS is lipidated. So this gives us what's called a positive control. When we put this KRAS GFP combined fusion protein in, this will show us what it will look like if the GFP protein is lipidated. And as you see, the cells look very different. You see if we zoomed in a little bit more, you'd see it's outlining the cell. They're not quite as bright. You see a very different pattern. And this reflects that protein being lipidated. So now the big question, what happens when we do the same experiment, but instead of using the known lipidated protein, KRAS, we use that 7B protein from COVID. And to our delight, we saw a very similar pattern. So what you want to look at is, this is GFP on the COVID protein. And notice that these cells, the fluorescence, the localization, looks a lot more like the KRAS version than the GFP alone. So this gives us confidence that this protein may be lipidated inside the cell. So it suggests the protein may be using lipidation. We have some other data, which working in collaboration with our a laboratory down at the University of Georgia in yeast models that also supports this, that I don't have time to talk to you about tonight, but it's really promising to keep pushing forward in this area. So we have these two questions. We're confident we're getting a check mark to that first one. The protein is modified. Now we need to know if it's important for modification for virus function. But the really nice thing is, if that second question is correct, we can jump ahead, because these lipidation modifications we study already have a whole bunch of drugs that have been in clinical development for over 20 years, actually as anti-cancer agents. But we can take all of these drugs, and then if we show 7B needs lipidation, we can basically cross-train them and see if we can put them in and see if they help out patients with COVID. So it gives us a way, what we call drug repurposing, take something that's already been developed and jump midstream. So we'll accelerate the ability to study the effect of these drugs in potentially treating COVID infections. Obviously, I have to thank the people who did the work. I mentioned Sadat here and Michelle, all of our collaborators. We've had funding again from the NIH, and moreover, especially thank the graduate students who are still in the lab every day at reduced capacity, really pushing this work forward. So that's sort of our little story of how things are going on in the lab in our COVID studies. And now I can throw it back to Lisa as we move forward through the presentation.
Great. One of the difficulties of this webinar style format is that there's no real applause, but I'm envisioning hopefully that all of our attendees are applauding like I am. I found those talks just fascinating and really exciting. And so now we're going to move on to the sort of second part of our program, which is really opening it up for everyone who's in attendance for questions and answers. And the first thing I want to say is when you registered, a lot of you had an opportunity to suggest questions that you were interested in ahead of time. And so the way I think we'll organize this question and answer session is, first, I'll ask a few of those sort of questions that you've already asked when you registered, questions you had about the virus and things related to the virus. And then in some, I'm looking at the Q&A bar now, and actually some of the questions are similar to the ones we already have. But please, as you're listening to our panelists answer questions, please feel free to post additional questions in this Q&A bar, because if you have a question, I guarantee someone else in the audience probably has a similar question and you're doing them a favor, okay? So without further ado, I will get started with some of the questions that you all have already asked in the registration. The first one is, I think, very relevant to what we heard, which is, what is the difference between vaccines, which we've all, of course, heard quite a bit about, and other types of potential therapeutic agents like nasal sprays? And in particular, of the research we heard today, which of those types of things is your research most likely to impact? So I guess I will hand that one to Professor Hougland first, and then we'll go from there.
Yeah, thank you. So really looking at it, and I got to say, I hadn't actually seen the nasal spray aspect of Ali's work, which is just completely awesome. It really is. So the difference is, right, that the vaccines are a way to train your body to fight the disease and to continue doing it, right? Whereas the nasal sprays are going to be sort of a one-shot protection. The way I describe it is the old adage of give a man a fish or teach a man to fish. Right, the nasal sprays are giving they'll protect you for a short time. You're giving a fish, so they'll eat for the day. Vaccines are the immunology way of teaching our bodies to fish forever, to feed themselves, right? And I think ideally, we want to have as many tools as possible. So going down both routes is really going to be our best bet to have a robust ability to prevent and treat COVID. So. That would be my perspective on it.
Thanks so much, Professor Hougland. Professor Schwarz, do you want to jump in?
Yeah, I mean, definitely this new information that's coming out with the Moderna and Pfizer vaccines for COVID are actually quite interesting, because this is a newly designed messenger RNA vaccine, which has never been used before on this sort of on any scale. So I think that in itself is very novel. And actually, just to give a shout out to another female scientist. So Ali mentioned a woman scientist who first discovered COVID. There is another woman scientist by the name of Katalin Kariko, who is a Hungarian-born woman scientist who actually thought about using messenger RNA to fight diseases 20, 30 years ago, and actually wrote a number of grants, and no. So she actually she was in academia, and she's now actually working with BioNTech, who's joined with Pfizer in developing their vaccine. So certainly I agree with Jimmy that a vaccine is you're training your immune system to recognize bad actors like the viruses, and so that your body will keep producing various antibodies, and your immune system will be able to address those situations, whereas a nasal spray is a one-shot deal. And I guess Ali certainly can better attest to, I think with the mentioned antibody, we're thinking more, it's the one-shot deal. So it be more along those lines as opposed to a vaccine.
Great. And also, if this is not my area of expertise, but it seems to me that also for some of the folks who are immune compromised where vaccines are problematic, the one-shot deal might be very valuable to provide them with some protection, which is, of course, why, it's again, just like masks, it's sort of a social good for vaccines, because then you're protecting people who might not be able to take them for various reasons.
Yes, that's an excellent point.
Very cool. Okay, so maybe we'll go ahead, because there's a lot of good questions. So I think we could talk about that one for a long time. But maybe, I think one of the questions that I found fascinating, just to give people a window into your lives, is how has the pandemic affected your ability to do your job as a scientist? Like, what are the challenges, and what have you learned? So maybe I'll throw that one to Professor Patteson first.
Yeah, so it's made the job a lot more challenging. I don't know what to say. As an experimentalist at a wet bench, you need to be in there to do your job. I guess, like Steve Jobs has organized Apple so that they maximize chance encounters from people from different areas, because there's a synergy that can happen. And that seems to be kind of taken away, because we're just not talking with people and chatting maybe in the same way. But I will say, though, that I've been really impressed with some of the creativity from my students. Actually, if I can still share screen, I will embarrass my students. My students have actually discovered that with virtual group meetings, you can pipette and do lab work. If you can see Max there in the camera, thinking that he wasn't being caught. So, you know, I think having a lot of flexibility is going to be really important, and we've learned the value of that.
Great. Great. So I'm actually seeing some interesting questions come up live in the Q&A box that are very related to the question I just asked before. So I think I'm going to jump to them because they're sort of follow-up questions to the ones we've already said, because I think they're concerns that are shared by many of us. So one question that came in the chat box, and I apologize to everyone in the audience if I mispronounce your name, but by Francis de Orio, is can these antibodies be produced in large sorry, the question just went away. In large quantities and in a cost-effective manner is pretty sure what I remember of that question. And so maybe I'll ask Ali and maybe Professor Hougland to address that one too.
I'll do my best. I think it depends a lot on what the antibody is and the production method. I know there are people whose sole job is just to figure out ways to produce these antibodies at a larger scale, which will help. And this is also why like having a strategy that depends on multiple different things is good, because some of them might be very cost effective. So they might be balanced by things that are, some might not be cost effective, and to balance those with ones that are.
Thanks, Professor Patteson. Do you have anything additional to add, Professor Hougland?
Yeah, I would say so. Production of antibodies is, I mean, these are large. They're a class of, you know, we call them biologics in terms of medicine, and they're more complicated to make in some ways than the small molecules, the drugs that like I learned that those of us who took organic chemistry learned about making, and we torture you with in Software Organic. But there's really been incredible progress in making these. For example, it was in the news a while back, especially a few months about the antibody treatments, like Regeneron has one that went on, got emergency use authorization. I remember that because I have an undergraduate in my laboratory who is currently working with them. I don't know about on that project, but there's been a lot of investment in ways to make these antibodies, make these biologic pharmaceuticals effectively and at scale. You know, they're not cheap, but as we do more, we learn more. So I think as we move forward, there's rapid development, and, you know, necessity being the mother of invention. If we need it, we're going to find a way to do it.
Thanks so much. And sort of, I think, a somewhat related question based on, you know, which is about the process of developing these vaccines and drugs, is a question by Jeffrey Stone, which is that all of the amazing work being done on possible vaccines at such a fast pace, is it realistic to be concerned about the safety of them, given that the usual time to market for vaccines could be several years, or even more than several years, if I can editorialize? Would one of our panelists be willing to sort of speak to that? I mean, none of us here are vaccine experts. That's not our explicit field, but since we have scientific biochemistry, biophysics expertise, does somebody feel that they could answer that? Professor Hougland? Oh, you're muted, sorry.
Oh, yep. I think that as I've been reading this, and this is something where, like I said, I'm not an expert, but I've been very appreciative with a lot of the work that's been coming out. I've become addicted to vaccine Twitter recently, especially people out of the CDC, Pfizer, a lot of public health schools. Vaccines come faster for a number of reasons. One, I feel the field understands vaccine and vaccine reactions a lot better than they were understood decades ago, so they know what to look for. Second, in particular, and this kind of goes to what Professor Schwarz was talking about the nature of these mRNA vaccines, my understanding, they're much less likely to cause some types of safety concerns because of the way they go in your body. Like, they're not putting an infectious reagent, like a deactivated virus, into your body. And so it's in some ways, it's much less likely to go in and really start causing issues. It still might, and that's why it's important to wait and measure. But then you always have to balance the cost benefit of what the safety, what the safety concerns are versus not having the vaccine out. I guess my understanding on the mRNA vaccines is the requirement was they had to have 2 months of follow-up safety data to see what the effects were, and what they're seeing are what they would say expected, right? But I think it's something they're going to keep an eye on, and it's something where I think the CDC, the FDA, they have a really good job, and their emphasis is to develop the safest vaccine and deploy as fast as possible. So I think that it's something they're going to keep looking at, but my sense of it, speaking just for myself, is that we're on a good trajectory and it's being done the right way.
Great. Oh, and Professor Schwarz has a follow-up.
Yeah, and just as I was mentioning before, I think there have been steps leading up to this that have been sort of 10, 20, 30 years in the making of initially this particular Hungarian-born woman scientist thinking about using messenger RNA to fight disease. And then you say, well, if I insert messenger RNA into the body, that could trigger an immune response that maybe you wouldn't necessarily want. And so what ended up happening is maybe about 15 or so years ago, the idea was to actually modify the messenger RNA so that the modified form could go into the body stealth way so that the body wouldn't know it's there. And then sort of once it enters the cell, do its thing. In this case, doing its thing means making the spike protein, which is these little spikes that stick out of the coronavirus. And so then your body, once you make them, then the immune system learns to recognize them and to knock them out. And so I guess the point is that this sort of research has been going on for tens of years, and it's now sort of being poised to be able to be used in an effective way. And the last sort of one other ingredient was to say, well, this modified RNA, it gets eaten up kind of easily by other stuff. And so they encapsulate it in lipid nanoparticles so that it doesn't get eaten up so easily. It can get to the cells before getting eaten up and do its thing. So there's been a lot of groundwork laid, and it's sort of the timing seems right. So it's not like it's just overnight, this is all happening. It's been, the groundwork is being laid over tens of years.
Yeah, thank you so much. I mean, again, this is something that I've been fascinated by because it seems in some ways we're really lucky that these mRNA vaccines were just about ready to be ready when the coronavirus hit, you know, in some sense. And they're really, if this works, it could revolutionize vaccines for not just coronavirus, but everything moving forward, because it would be a real step forward. So it could be one of those breakthroughs. I mean, we got very lucky that its time has come, you know, there's still work to be done, still things to say, but that's one of the reasons I think those of us who have read the literature a little bit are hopeful that it makes sense, that nothing has been rushed in some sort of bad way as far as we can. And it sort of makes sense that maybe this is we just got lucky in that they're ready now. Is that fair to say? Yeah.
I think somewhat, yes.
Great. Okay. So I think maybe I will continue because we had a little bit of a chance to talk about how the pandemic affected your labs. And I'd be interested if either Professor Schwarz or Professor Hougland would like to address that question. And in particular, I think also thinking about, a lot of you mentioned, you know, the heroic work that your students and postdocs have been doing. And we heard about that a lot. So do either of you have any comments about sort of how those personnel things have been going in the pandemic? Okay, Professor Hougland.
Sure. Yeah. No, it's funny. I had a group meeting today at about 1 o'clock, and we spent about a half hour talking about this. It's been challenging, but I've been fortunate that I'm working with a lot of really dedicated young scientists, and we figure out a way to do it. I will say, back during the summer, SU was closed. Basically, the labs were all closed for a number of months. And one thing we had to do is we had to write up a reopening plan to even bring anybody back into our lab, like, you know, beyond myself and my research technician to do basic maintenance. The number one watchword in my lab has been safety. And I've been talking to my students a lot about this, that if you ever feel unsafe, if you're not sure about distancing, like, walk away and we'll figure it out. But the benefit we have, particularly in my lab, we do a lot of cell work. I'm fortunate that my people have been trained to deal with biohazards. And so the training we have for that in our research has translated very nicely, almost frighteningly so, to the real world right now. Okay. But it's something where I see my job right now to provide them as safe an environment as possible, bring them the resources. but also part of it is right now is reassuring them because research is not going as fast as it normally does. It's slower. There's breaks. Oh, I gotta say, I love the pipetting during group meeting. That's awesome. But letting them know that's okay. Take care of yourself and keep the research moving forward, but do it in a safe way. And it's important that we're moving forward. We all have adjusted to the pandemic world, and we just keep working through that and making sure that, I look to, am I supporting my people as well as I can so they can continue being productive in research and being, sort of healthy in their everyday lives.
Great. Thanks so much. So I want to be respectful of everybody's time. I see we have maybe about just like a few more minutes left in our scheduled session. And I'm going to try to end this as, given all of the challenges of the pandemic, I want to make sure to try to end this as close to the stated end time as possible, because I know you all have other things that that need your time. But I think one of the things, so we've gotten so many good questions, and it's going to be hard. We obviously probably can't get to all of them, although we may be able to stay on for a few extra minutes to talk to individuals. But I think one of the things that has come up a lot in the questions, both before and after, is a little bit more about masks. Because we already heard from Professor Patteson, a little bit about the physics of mask wearing. And so I think one of the questions people have is, what adjustments could you make to masks to make them more effective? So for example, would a hydrophilic mask do a better job of catching droplets? Why not treat the masks with some of your stuff that would go into your nasal sprays as well? Like, could you have like a treated mask or something like that? So maybe I'll throw that one to Professor Patteson, if you want to comment.
Sure, I can try. Yeah, it's not a crazy idea to think about trying to treat masks or coat masks with different drugs that will capture the virus. Although maybe I mentioned the masks are really more capturing the respiratory droplets versus the virus themselves. So I really, one critique I hear people say is, I can wear a mask and I can get sick anyway, so why should I wear a mask? But I think, that the idea here is really about the probabilities, right? Like I showed the pictures about how much load you can get with and without the mask, even just kind of a cotton mask. And I think it's pretty incredible how much you can cut down the spread. So yes, you still possibly can, but it's a 1 in 100th chance of every possible interaction. That and, social distancing and washing your hands are most effective.
Thanks so much, Professor Patteson. Okay, so what I think I'm going to do now is sort of wrap up the official version of this forum with just a few brief comments. And then I see we have a few questions, and maybe I would offer that the panelists could stay online for maybe a few more minutes, like 5 more minutes, and see if we can sort of tag team on some of these questions that people have. Because, the nice thing is you can also type answers to some of them. So maybe we can provide, that's one of the good things about Zoom and all of these tools that we have now, is you can do some cool things you couldn't do in a real in-person lecture. As we close, I just would love us all to give a round of applause, into the void for our, our really fantastic panelists. It's one of these things that sometimes you're so proud to be associated with the place because you're associated with people like this. And that's the way I feel this evening, having the pleasure of moderating this with these obviously brilliant people who are still real world focused, and trying to really help people. And I want to thank all of you, because I think one of our best chances of, making the best out of the situation is to pay attention to science and think about the facts behind the science. And I can tell by your participation here that you're all doing that. And, you're, in my opinion, you're to be commended. And I really appreciate you taking the time out of your very busy days to spend with us this evening. And so the BioInspired Institute is actually planning to hold a few more of these public lectures in the spring. And so we'll be sending out information about that. And we'll also be sending out an assessment to see what you liked and what you didn't like about this format. So if we do it again, we can do an even better job. So thank you again so much for joining us. And we hope to see you or interact with you again soon. Thank you. So everyone feel free to sign off. And then those of you who stay, I think for the panelists, maybe we could look together at this Q&A and then we can type some answers. And so maybe we'll do this a little asynchronously for those of you who think you can address one of these questions. We can try to address some of them as best we can. Yes. Okay. So I think we can all just click and start typing. The other thing is that if one of us wants to take a question verbally now, like we could type while somebody answers a question in real time. Does anybody want to take one of these questions in real time? Professor Hougland?
Yeah, I just almost laughed in a really great way. The question from Robert Hallenbeck: both vaccine trials yield surprising results in terms of effectiveness. In your own labs, how would you respond to such surprising results? The first thing, my students crack up because I say this all the time: never show me one result, show me three. Right? Reproduce. And the thing is, the vaccine trials they're massively reproduced. But yeah, that's the fun of it, though. I joke with my lab, my favorite quote from Isaac Asimov is, the greatest discoveries in science aren't greeted with eureka, they're greeted with, that's funny. Like when you have that unexpected, surprising result, that's where some of the fun is. So yeah, I'd be suspicious of it in first, but I'd want to go back if it held up to a reproduction and controls, then you run forward with it.
Great. Does anybody else have a question that they would be able to respond to in real time? We're still figuring out how to do this, folks. So thanks for sticking with us.
Yes, I will type in the name for Albert Lennell. I will type in the name of the woman scientist. So I wrote the name down. So Catalin Carico. Okay, I will do send. I guess, David Zach, should I answer that question?
That would be fantastic.
Is it believed that the vaccine will specifically block the virus, or rather the sequel of the virus, such as blood clots, whatnot? So the vaccine will the vaccine will cause your body to produce some of those spike proteins that are on the outside of the virus. And then your immune cells, your T cells and whatnot will actually recognize those spike proteins as bad players and knock them out. And so basically the vaccine is training your immune system to deal with these spike proteins, which are what are necessary for the virus to get inside the cell and replicate.
Right. All right, so I think we're well on our way to answering a lot of these questions. It's about 5 past the end time of our lecture series. So I think at this point, you can always, our information is publicly available on the Syracuse University website. The BioInspired Institute has an email address and a website, so feel free to reach out. We're always delighted to help explain the things that we do. We could talk about science all day, and that's not always a good thing, but in this case, maybe so. And I think we'll close there if that sounds good to everybody. Thanks again so much. Have a great rest of your evening and stay safe. Those of you in Onondaga County and surrounding counties know that it's an especially important time to be careful and stay safe. So good luck.
Thanks, Lisa. Thanks, everyone.
Thank you.